So today, we're going to dive into a real medical mystery. It's a story that connects a single tiny genetic flaw to a nation's health. We're going to follow the clues, starting with a really surprising cancer cluster, and trace it all the way down to a single letter change in our DNA, and then see how that incredible discovery is actually saving lives right now.
Okay, so the whole story kicks off with a massive puzzle. There's this very rare childhood cancer. It's called a pediatric adrenocortical tumor, or ACT for short. I mean, this thing should be almost unheard of. But for some reason, it was just popping up again and again and again in one specific part of Brazil. You know, this wasn't just a coincidence. It was a huge red flag that something was going on.
And get this, the scale of the problem is just staggering. We're not talking about a little statistical blip here. The rate of this cancer in southern and southeast Brazil is 8 times higher than anywhere else on the entire planet. An anomaly that big? Well, it demands an explanation.
All right, to get to the bottom of this, we need to go microscopic. We're going to zoom right into our own genetic code and meet one of the most important genes in the human body. It's a gene called TP53, and its job is so critical that it's earned this really heroic nickname.
It's known as the guardian of the genome. I mean, think of it as the ultimate quality control manager for every single one of your cells. When your DNA gets damaged, and that happens all the time, from things like the sun or just simple mistakes when cells divide, this protein, P53, steps in. It slams on the brakes and stops that damaged cell from turning into cancer.
Now, this guardian protein isn't just some simple blob. It's actually a pretty complex machine with different functional parts, or domains, as they're called. For example, it has a part that lets it literally grab onto our DNA, and this is going to be really important for our story, it has something called a tetramerization domain. This part allows 4 of these P53 proteins to link up together, and that 4-part structure is absolutely essential for it to do its job.
And believe me, this guardian is a serious multitasker. It doesn't just do one thing. It can pause a cell's life cycle to buy time for DNA repairs. Or, if the damage is just too bad to fix, it makes the ultimate call. It triggers the cell's self-destruct sequence. It's a process called apoptosis. It's basically the cell's hero.
So you can see the stakes are incredibly high here. On the left, in a healthy cell that's under stress, the guardian P53 kicks into gear, and everything gets back under control. But on the right, when P53 is mutated, it just can't do its job. That damaged cell is left completely unchecked, free to keep dividing and potentially grow into a tumor.
So here's the crucial point. When someone inherits a faulty, broken copy of this guardian gene from one of their parents, it can cause this devastating condition called Li-Fraumeni syndrome. And this puts them at an extremely high risk for developing all sorts of different cancers, often starting when they're very, very young.
And that brings us right back to Brazil. When investigators started analyzing the DNA of the children who had this rare cancer, they found their primed suspect. And it wasn't just a random collection of genetic mistakes. No, it was something far more specific.
This is where it gets really wild. They found this incredible consistency. More than 90% of the kids with this rare cancer, all of them, carry the exact same mistake in their TP53 gene. This was the breakthrough. This was the genetic thread that tied all of these cases together. And here it is. This is the genetic fingerprint. In a massive gene made of thousands of letters of code, the error was pinpointed to one single spot. One letter, a G, was swapped out for an A. This one specific change is so famous in genetics, it even has its own name, R337H.
So what does that tiny DNA swap actually do? Well, it causes a one-for-one swap in the protein's building blocks. An amino acid called arginine, that's the R in the mutation's name, gets replaced by another one called histidine, the H. And this one seemingly tiny substitution was the smoking gun responsible for this entire cancer cluster.
Okay, so the scientists had their culprit, the R337H mutation. But that just raised an even bigger question, right? How did this one specific, very rare mutation become so incredibly common in this one part of the world?
Well, the answer lies in this really powerful idea from population genetics. It's called the founder effect. Basically, it's what happens when a brand new community is started by just a handful of people, and by pure chance, one of those original founders happens to carry a rare genetic trait.
And that's exactly what genetic analysis points to here. You can almost picture it, right? It seems that many, many generations ago, a single person of Portuguese Iberic heritage who was carrying this R337H mutation settled in Brazil.
And as they had kids and their kids had kids, that mutation was passed down, spreading silently through the population over hundreds of years.
So you fast forward to today, and the result is that about 1 in every 300 people in this region of Brazil now carries this specific mutation. This takes what is an exceedingly rare genetic variant everywhere else in the world and makes it a relatively common and serious public health issue there.
All right. This next part is, for me, probably the most fascinating piece of the whole puzzle. How this specific mutation actually works to cause disease. Because, it's not as straightforward as you might think.
See, most of the TP53 mutations that cause cancer are kind of like a wrecking ball. They just completely demolish the protein's function. But R337H is different. It's more subtle. It's like a hidden structural weakness, a flaw that only becomes a problem under very specific conditions. Hey, remember that tetramerization domain we talked about earlier? That 4-part structure? Well, the mutation sits right in there. A normal P53 protein assembles into a nice, stable 4-part structure, a tetramer, and that's how it works. But with the R337H mutation, that structure becomes wobbly, unstable. And its stability is super sensitive to changes in the cell's acidity, or pH level.
And this was the big aha moment for the researchers. They had discovered a totally new way for a mutation to cause cancer. See, it's not that the guardian protein is always broken. It's that it becomes dysfunctional. It fails under certain stresses that change the pH inside a cell. This conditional pH-sensitive flaw is the reason for the cancer.
This incredible scientific detective work, it wasn't just some academic exercise. It did more than just solve a mystery. It paved the way for a direct, life-saving public health solution. Understanding the problem was the critical first step to actually fixing it.
So, here's what they did. Knowing the cause and, crucially, who was at risk, they created a proactive screening program. Newborns in the region are now tested for the mutation. If a baby carries it, their parents get counseling and are taught the very first signs of these tumors. The children are then watched very closely, which allows doctors to take action immediately at the first sign of trouble.
And the results of this program? Honestly, they are nothing short of astounding. When this cancer is caught late, it can be really tough to treat. But when it's detected early through this surveillance program, the cure rate is nearly 100%. It's just a perfect example of how deep genetic knowledge can be translated directly into saving people's lives.
The story of R337H is just this powerful reminder that deep inside our genetic code, we can find not only the roots of a disease, but sometimes also the keys to preventing it. It really transforms our view of cancer from what can feel like a random tragedy into something that might be predictable, and in this incredible case, something that is now preventable.